listeners, Yamina here. Welcome to the Dr. GPCR podcast. This conversation has been 3 years in the making, and I'm delighted to welcome Dr. Ajay Yekkirala, co-founder of Superluminal Medicines. When we recorded this episode, Ajay and I were colleagues at Superluminal, which made this conversation about science, innovation, and entrepreneurship especially meaningful. In this episode, you'll hear how a childhood fascination with space grew into a lifelong quest to understand GPCRs, how mentorship from scientific leaders shaped Ajay's values, and how asking the right questions led him from academia into biotech entrepreneurship, building companies focused on targeting GPCRs. We'll also explore his latest venture, Superluminal, where AI meets GPCR dynamics to unlock the next generation of medicines. But before we begin, a quick reminder, Terry's Corner is live. Every week, Dr. Terry Kinakin breaks down complex pharmacology into clear, practical insights in bite-sized courses and live monthly Ask Me Anything sessions to go even deeper. I'd also like to take a moment to thank our partner, Celtaris Research, for their support. They help accelerate GPCR drug discovery with innovative chemical tools. You'll find all the relevant links, including our featured partner, Celtaris, in the episode description. And now, let's dive in. Hello, everyone. This is Yamina from Dr. GPCR, and today I'm very, very excited. This is a project that has been going on for over 3 years now. I finally got my friend Ajay Yekkirala on the podcast. Ajay, welcome.
Thank you, Yamina. I know it has been a long time coming, so sorry it took this long.
Hey, as long as it happens, we're going to be happy with that.
Absolutely. I'm excited. Thanks for having me.
Absolutely. It's so good to have you on. So let's start at the beginning. I think with the disclosures, you and I met 4 years ago, actually. Our first meeting anniversary is coming up because I think it was sometime in January 2021.
Yep.
On a panel discussion about screening to find GPCR ligands.
Yes. I think it was Annette, right? Like it was her.
Exactly. It was Lab Roots, Annette Jill Christ organized this panel discussion, and we kept in touch. And for over a year now, we have been working together, and it's been a fantastic ride so far.
No, thank you. I mean, it's been great having you as well. And, you know, it's remarkable how what seems like random meetings end up being such lasting relationships. It just really shows the power of meeting people, networking, and also however large biotech is, it's still small. And I think it's again a testament to that.
It is a very, very small world. So I know you and I know each other. I'm going to pretend I don't know you that well. And I'm going to start by asking you, can you please introduce yourself? Tell us who you are and what do you do?
Absolutely. I'll also try and pretend. This is going to be hard. All right. Hi, everyone. Ajay Yekkirala. My background is that I'm trained in pharmacology. I spent my early training career as a graduate student with Phil Portoghese out at University of Minnesota, Twin Cities. And Phil, is this Hall of Fame chemist. When I joined his group, I think he was already almost 50 years in the field, had generated, many of the opioid antagonists and agonist ligands we've heard of. Just an absolutely phenomenal scientist as well. I learned a lot from him. I owe a lot of my own scientific curiosity and research methodology to him. And I think as we typically are trained to do in academia, you are trained to be again in academia. And, given, my training with Phil, I was very keen that I'll become a PI. And so I wanted to do a really good postdoc and again, continue on that journey to become a better and better student of science, if I could say that. So that's when I moved over to Boston Children's Hospital, Harvard Med, to work with Cliff Woolf in the neurology, neuroscience departments. And that was a fantastic experience as well. A really great mentor. And, I got to kind of appreciate how to think big vision ideas without any breaks on your mind. Just, how big can you really think and how wide a net could you cast? And I think Clifford is one of those, visionaries. So I think it was really great that I, got to experience that as well. And as I mentioned, I was actually, I'm a third year, somewhere around 2014, third year postdoc. I started interviewing for postdoc positions. I was even getting offers actually at different places. But then I had this midlife postdoc midlife crisis, I guess, or I don't know how else to call it. But basically, I started like wondering why I was in it. What actually drove me to science? Is this what I want to do? And I really love doing science in academia. Don't get me wrong, and I think I would have been very happy doing that. But I felt I owed it to myself to kind of think through that a little bit more. And one thing led to another, and I suddenly decided to follow an entrepreneurial side of things. And that came from, a few things that come together. A couple of my colleagues, they were a lot more entrepreneurially minded. And they were tagging me along to Harvard's innovation lab, all these different entrepreneurial meetings and, ex-alumni coming in and giving talks about what they did, when they started out companies. And so that kind of, intrigued me a little bit. The second thing that ended up happening was I realized that there's this little graveyard period where fantastic bench science in academia, we hope, gets actually picked up by pharma. But typically that doesn't happen because there's still a significant amount of development work that needs to happen before any asset looks promising enough for pharma to step in. And certainly that was the case, like 2014, 15, when we started to think about this, that became a graveyard zone where really good academic science, very promising assets, but then no one knows if there are toxicity issues. No one knows if it'll actually fly through enabling work, So I realized, maybe the way to do that was to actually start a company around certain assets and then maybe build those out and fill up the space along that, do true translational work. And this jived well with me because I came to science wanting to develop medications for patients. And I felt this would be an opportunity for me to test drive that. So that led to the formation of Blue Therapeutics, the first company I founded. And then rest is history, I guess. I can talk about all those things, but yeah, just as an introduction, just to give you a sense of where I think.
We can get to that because I have a bunch of follow-up questions, but let's start at the beginning. Did you always know you were attracted to science or you were going to be a scientist? Like 9-year-old Ajay or 10-year-old Ajay?
Yeah, it's such a great question. I was always a very curious child, and I guess many kids are because they're always asking questions. But I always found, I think the first real dream I had of what I wanted to become, and I recall I was like 4 or 5, and I wanted to become an astronaut. Like, I think I'd seen one of the space shuttle launches or something. I can't recall what it was that I saw, but it was so inspiring. And I'm like, wow, to be amongst the stars and go to the moon and, all that. So there was always that sciencey nature to, I guess, my dreaming.
Yeah.
This is like all past the Spider-Man, Superman stage, right?
Yeah, of course.
And I think, medicine and health, in my family, parents, a few others had gone through some really significant health issues when I was a kid as well, which also made me kind of really gravitate towards health and healthy living significantly. So I think like all those things kind of got married together, and I was probably about 10 years old when I knew I wanted to be in the healthcare, side of things. I think I pretty much had made up that mind. Whether I would be a physician or a surgeon or a scientist, I really hadn't fully baked that, I guess, at that time. And then it took me several years because even before starting grad school, I was actually thinking of going to med school. Even had gotten into med school, but I decided along the way that my scientific curiosity I owed it to myself to kind of give it wings and go down the path of research rather than going to medicine proper.
I love that. So I don't think I've had anyone said I wanted to be an astronaut. So it's a first in the podcast, ladies and gentlemen.
Yeah. In fact, you will laugh. Like when I started thinking about research, I was looking at astrobiology to see if I could kind of sneak into NASA that way.
I love it. I love it. Yeah, and a lot of guests also in the past mentioned, oh, they wanted to go to med school, and they started, or pharmacy, or all of these things, and then they ended up gravitating towards research. I remember you told me the story on how you got into your PhD lab, and I think that's worth sharing with the audience.
Sure, absolutely. I will gladly share. So as I mentioned, my scientific curiosity part, I knew I probably wanted to follow that. This was the early part, as I was wrapping up undergrad. And I thought maybe I should, give some credence to both of those ideas, being a physician and doing research by going after these physician scientist programs like the MD PhD program. So I applied actually to MD PhD programs in several schools. And unfortunately, I didn't get in anywhere. But at University of Minnesota, when I got this really interesting email from someone who was on the admissions panel. He was a professor in the neuroscience department. He reached out and said, "Hey, Ajay, I just was intrigued by your profile. I thought it was really good. This year, we're actually not taking any, MD PhD students because of some funding issues. But maybe you can, bolster your profile further by doing research for an additional year, because that seems to be, one of the drawbacks in your overall profile. You probably want a little bit more research." It was so nice of him to do that. I'm not sure what actually drove him to do it. To date, I'm very grateful to Dr. Lowe for doing that. So then I wrote a note back to him saying, because neuroscience was always something interesting to me. So I asked him if he would recommend any groups where within that department that I should be, reaching out to. So then he actually forwarded my CV to the neuroscience department at UMin. And then a few days later, I get this email from this distinguished professor of medicinal chemistry. And I'm like, okay, how did it go all the way to MedChem? And I didn't know anything about Phil Portoghese at that time. And so I Googled him, and I was like, he had, he had, I think, like 500 papers or something at that point. I was like, wow, this is a true stalwart of the field. And then I came to realize he was in the chemistry department, medchem, pharmacy, neuroscience, pharmacology. He was pretty much, on every department, I guess. Anyway, so Phil reached out and said, "Hey, I have this lab tech, position. I found your CV interesting. Do you want to come join?" So I ended up working with him. I really loved it. Like just my first meeting with him itself, and he offered it to me right away to join the group. And I said yes right away as well. And then it was a match made in heaven. So I did that, and I was still keen on applying to an MD PhD program. And I wanted a recommendation letter from Phil, so I talked to him about it 6 months in after starting in his lab as lab tech. And then he told me, he said, "Oh, MD PhD, like, why do you want to do that? It's like a long program. And in the end, you will either focus more on being a physician or a scientist. So pick one now." And I was not convinced. And so he had several MD PhD students who had come out of his lab. So he introduced me to all of them. So I ended up talking to 5 or 6 of them. And every one of them echoed exactly the same thing. They said, "Yeah, it's nice that we got exposure to one or the other thing, but we could actually do without that and still be in the same place." So taking that, and I had already started applying to plain MD and plain PhD programs as well, just to make sure I didn't get into any MD PhD program again.
What did they have against you?
Meant to be, yeah. It was just, not meant to be. But then I did get med school offers. And then I thought, let me roll the dice, because I really enjoyed working with Phil, and I realized I was working with someone truly at a different level. So I'll tell you this funny story. I went to a very famous department in an Ivy League school, who shall not be named, but they told me the only reason they were talking to me is because Phil Portoghese wrote me a recommendation. And I started thinking through that, entire day of interviews there. I'm like, if that's the case, then why don't I work with Phil Portoghese? Right? Like, what's the point? So that's when I came back and I made an offer to Phil saying, "Phil, I will become take a pure PhD program if you take me on as a student." And he was taken aback because he was already, in his late 70s at that point, and I think he had considered maybe retiring. But just to tell you, Phil retired last year at 92. Okay, like this is not someone who would have retired. It's only because of health issues that he has now. But but he said, like, look, you joining the group has also rejuvenated me. So maybe this is something we should do, And then so he, also wanted to do it, and that's how I ended up
I
in the
love it.
PhD program. Yep.
I love it. How many papers did he end his career with? Because he had,
Yeah.
20 more years in there.
Yeah, I know that at least after I left in 2011, probably another 50, 60 papers, at
Wow.
minimum. Because it's just, he's a very prolific,
Yeah.
scientist,
Yeah.
full of ideas. I'll tell you, the most amazing memories I have being in Phil's group, where I would go into his office with an idea or a question, and 4 or 5 hours later, sketching a whole bunch of things on the whiteboard, I would come out with this, and having read an encyclopedia. That's how his brain worked. It was an encyclopedia of knowledge and understanding. So time would just fly. Like we would just go through 5 hours of a discussion. So I really miss that, and I credit him for taking that time out to mentor a student, even though he was so senior and he had like 11 or so postdocs, I think, when I was in the group. So he was a busy guy. He was also editor-in-chief of JMedChem. I mean, he had so many things, but he gave time, to mentor, which, remarkable.
Yeah, and I think he was the way you talk about him. I think he was teaching through showing you how his brain works, how he thinks, and
Yes.
that is absolutely critical because you're coming to the lab, as a fresh PhD student, you have no idea what's going on.
Yeah.
And if unless you have somebody showing you the ropes and showing you, hey, this is how I think about it. How about you learn this and then you can form your own ways? But it's, you start off with a good foundation.
Yes. I because you said this, I will tell you the story. You know how typically PIs come with ideas, and then you execute on some of those ideas, and then they let you go on your own to develop those. Phil was the exact opposite. He'd be like, what is your idea? Where do you want to go? Like, in that sense, what a mentor. And I remember I had generated data that actually negated one of his papers from like 1982. And I was worried about taking this to him because I was like, hey, is he, yeah, like, is he going to just be like, what, this is wrong or whatever? I take it up to him. He immediately said, oh, this is really interesting. And he looks at it and he's like, wow, this is fantastic. This means we were wrong in 1982, and just that level of scientific integrity has stayed with me. He taught me how to really do science with integrity and curiosity, right? Like that was just didn't bat an eyelid. He's like, yeah, we were wrong. We got to fix that. And we need to put this out, right? Just that level of clarity.
Yeah. And I think it's unique as well. Not everyone has that integrity. Not everyone has that, level of all that matters is the data. And is it a well-controlled, well-run experiment? Do we have a good hypothesis? And whatever the answer is it a yes or no? The data is the data.
Exactly. It's always that way. Because we're sharing about this, like I'll tell you one more story. After I left Clifford's group, there how like some of these papers get left behind, and there's people who then come on later who try and wrap that up for you. I was working on a paper, and it was pretty advanced, and we were ready to submit it. And we wanted to do a few additional experiments. The new person who came in, they generated data that completely negated about 3 years of work that I'd done in the first group. But I was like, hey, we were wrong, and it's fine. It's okay. It's okay to be wrong, right? I think that's the biggest lesson, I keep kind of reminding myself of.
Yeah, no, I agree. And I think sometimes whenever the more technologies become available, the more tools we have, the better we can get a sense. It's like, changing lenses.
Yes.
And that's also important. I'll say it sucks because it's 3 years worth of your time and your work.
Yeah.
But at least you have your answer.
Exactly.
And it's important.
Because at the end of the day, we are just trying to progress our understanding of science. And I think you said it very, very correctly. As you get new technologies, you're able to look at the same problem a different way. with probably more robustness, right? Like as technology's improved, we only got more robust. And it can sometimes, like maybe something that was an outlier look real just because of the tech we were using at that time. And that was exactly the case, in both cases that I've mentioned, the both stories.
I love it. I love it. You mentioned something while we were mentioning how you got into Clifford's group and here in the Boston and at Harvard about learning how to think without boundaries.
Mm-hmm.
And that's an important point. I want to get back to it a little bit because I think, depending on who you train with, and I don't want to generalize, but a lot of times I've seen it, and I have been like that as well. And it's something that I've been working very hard to change is you're so focused on the details and you're so focused on what cannot be done that it's very difficult to think about what if it can be done. And we'll worry about the details later.
Yeah.
And I think that's that space where the innovation happens. So tell me a little bit more about how did this type of thinking help you in your career and define the next steps?
It's such a great point. And I really wanted to reflect and I guess to use the fat term today, double click on what you just said.
Ooh, I've never heard that one. That's a good one.
Yeah, it's apparently this finance bro thing that I thought I'll throw it into our jargon. Anyway, you said it beautifully. It's only when we start thinking about not what cannot be done, but if it could be done, what would we do? And I think you said it beautifully. That's where innovation comes to bear. I think I've actually seen that from personal experience, even within what we are doing, for instance, currently at Superluminal and in the past, and also even outside, like how innovation kind of moves. Everybody thought you couldn't go to the moon. That's why it's called a moonshot. Like the whole concept of a moonshot is you mostly will not make it, right? But we did. And I think, that's the beauty of it. So the question then that I was taught by Clifford and Phil to really think about, they may not have framed it this way, but this is how I've understood what they tried to teach me, is don't think about what you cannot do or, what's the limitation of the current technology, but really ask the question scientifically, what is it that you critically want to learn? So if you define your question on trying to peel back, a little bit and peer over that parapet wall of current understanding, the only way you can do that is actually through some significant inflection of idea, technology, thought, all of that baked in, right? Fundamentally, you cannot, ever limit yourself because the questions are too many. Like, in fact, there are many questions that we still don't have answers for. So I think it's great that there are so many questions, right? How is memory formed and stored? Such a fundamental thing to human life.
Yeah.
And we still don't fully understand it, right? Like there's so much more to explore. So I guess the way I've therefore started framing questions for myself is, we are here today for a particular topic that let's say we're trying to develop a drug for
Or GPCR, obviously.
yeah, for a GPCR, right? And let's say we're going after depression or pain. Let's talk about pain. Blue was that anyway. So the biggest issue is something about our understanding of the circuitry of pain tells us that if you try and take the pain away, you can do that. But also, most of the time, those pathways would also impinge on other circuits. And that's how you get side effects. Now, what's the issue with the opioids? You can get fantastic pain relief with the opioids, but the opioidergic neurons will also, project into the limbic system. They're projecting into the pre-Butzinger complex of the brainstem. So that's where you get respiratory depression. With the limbic system, you get reward, blah, blah, blah. So one way to think about it is, hey, this is the limitation. Like if I try to take pain away, those pathways are impinging on this. What am I going to do? I don't know how to take that out. Or the question is, maybe a reframing of that is, imagine if you can actually dissect the circuits of pain so minutely that you're only be able to hit that, and you're not actually hitting the limbic system or the pre-Bozinger complex. Now, if you could dream and figure out how you could do it, so you have to ask that question, right? So the more fundamental that question gets, then it makes you think about, how do I then tease apart this? How do I actually break apart those circuits? And you might say, technologically, based on what we have today, it's not possible. But because you have asked that question, you will go down that path, and you will find an answer, right? And I think that's where innovation can happen, as you said. Hopefully that gets at the core of it.
I think it does. And I think, going back to any circuitry, I'm thinking here about pain. Pain is important. Exactly. You need, if you don't feel pain and you put your hand on, in the fire pit that we were talking about yesterday, then you're going to burn.
Yeah.
Then again, when you think about it and you think about GPCRs and balance in general, maybe the one other way to think about it is, can we diminish pain enough so that the side effects are not as high or as, life-threatening?
Yes.
Because you're maintaining that balance. But to your point, if you don't ask this question, then your brain is not going to even think about, okay, this is what I want to get to. I want to answer these questions. What are the tools that I need to create to be able to then answer that question?
Absolutely. And I think, it's very nicely said, right? Because most of the time, what we don't want is there for a reason. Evolution didn't choose it because it made a mistake. Like, I'm not saying that there's a big intelligent design for evolution, but evolution conserves things that help you. Pain is a cardinal sign, as you said beautifully. Like, if we didn't feel pain, you would probably die in infancy because we would do something to ourselves, right?
Yeah.
And so that's the reason why pain exists. So, just to kind of belabor on this, we realized there are two problems. One is pain gets centrally sensitized. This is the whole thing that made Cliff Woolf famous. The whole idea is it gets so sensitized centrally that no matter what you do in chronic pain situations, the central pathways are just overactive. So the peripheral mechanisms are not going to help you. You can block pain at the periphery, but it's already sensitized here. So even a small change is going to make a bigger impact. So that's one problem. The other problem is early acute pain, you can block at the periphery. You may not need to actually hit something centrally. So that's the one thing people have thought about. It's like, what if I hit trip channels? What if I hit those kind of things, or P2X4, whatever. So the whole idea is to be able to separate that for acute versus chronic. We thought maybe the way you bridge these two is if you actually hit something supraspinally, so something in the spinal cord, and not supraspinally in the brain. So if you just do that, so if you can find a target there, you're not going to have activity centrally. So you're not going to have the limbic, pre-Bozinger issues, etc. At the same time, it's going to be better than just blocking acute pain. And that was the theory that we worked on at Blue, right? And I think we found a target there, and actually it worked beautifully. No respiratory depression, 100-fold better potency than morphine, etc., etc. But we would have never gotten there. We wouldn't even look at that if we didn't think about that, right? Like in terms of just separating those out. I think even the Superluminal story comes together exactly in the same way. Everybody, and maybe, I'll let you kind of maybe guide us.
We can get to that in a second. I want to go back. I have two questions for you, which are the hallmark of the podcast, and then we can talk about the Superluminal story, and I'll put in my spin around that. So question number one, what is your favorite GPCR? It's a tough one, right?
It is a tough question, but I do, I have, so at different times I have had different favorites, but if you ask me at the heart of me, what I think about as a quintessential GPCR, it's the opioid receptors. Reason is they're the best studied. We've learned a lot about GPCRs actually through the opioid receptors. And something about it just makes me feel, they're named, the ligands are named after Greek gods, like Morpheus. So I'm like,
The
there's
king of GPCRs.
yeah, it is. I feel like, it is at the pinnacle of the research aspects of it as well. Yeah. So that that
I
that
love
would be
it.
my answer.
I love it. All right. Next question is, during your graduate studies and postdoc, when you obviously started working on GPCRs, was it for you a kind of an aha moment of, oh, this family of receptors, or was it more this receptor or this group of receptors or GPCRs in general? Because I've spoken to a lot of guests, and sometimes people said, oh, it was the family in general because of the diversity of signaling. Others said, well, actually, I didn't care. I just came in and this is the receptor I started working on, and I, get them in a series, in a production manner. What was it for you?
It's a fantastic question. My first exposure to receptors in general and GPCR specifically, I think, was in a neuroscience class in undergrad. And the thing, obviously in class they don't go that deep, but the thing that did fascinate me was just signal transduction cascades, how they worked, right? Yeah. So
Same here.
you can relate to that, right? And I'm like, wow, you can have a small effect that then cascades into bigger and bigger and bigger, depending on the handoffs that happen along that pathway. And it just like blew my mind one day. And this was, I think, probably as I was doing research in Phil's group, that there are 800 something GPCRs, if you throw in the olfactory ones as well. But there's only a dozen or so G proteins. And it started making me wonder, like, wait a minute, how is such complex biology, significant selectivity, many of these having their own native ligands, yet we are able to transduce them through the same, so they're all shared, right? So in a given neuron, you could have several GI coupled receptors all signaling at the same time, and you could still separate out in some way an opioid effect from a different effect. So this like just blew my mind. I still don't understand it. Okay, like, and I've asked a lot of people, and I don't think anybody still gets it. There's a lot of theories. Okay, we can discuss that later. But that, I think, drove me to GPCRs as a class to say, OMG, if there's a way, there has to be some level of conservation, for even such a complex set of receptors and mechanisms being transduced with such a small number of G proteins. Yes, with bias signaling, we have the arrestins, we have all that, but still, we don't have 800 transducers.
Exactly.
For a fact, right? So there's still significant conservation there. That, I think, remains the reason why I still am like fascinated. I feel like we want to crack that code. Like, what is that code? How does toggle switches in a GPCR pocket work? Allosterically, how do things get transduced? Even biophysically, I mean. There's so many questions. It's just exciting. We could spend 15 lifetimes and probably still not understand these.
No, I agree. And then it's interesting because you put it so elegantly, and that I feel exactly the same way. I have tears in my eyes because that's exactly what fascinated me. You have a limited number of G proteins, a limited number of G protein subunits, and you have all of these receptors.
Yeah.
That can be co-expressed with the different G proteins, the different subunits. They can do all sorts of things, and then yet there's humans actually thinking about this thanks to GPCRs.
Yes.
It's just phenomenal.
Mind-boggling. I'll mention this one thing because I think the listeners might find it fascinating. When you think about peripheral neurons, like all the peripheral neuronal cell bodies, they all come together into the dorsal root ganglion. So these DRGs are right by the spinal cord. They sit right outside of the vertebrae, right there in that the little pocket. And they project these long axonal projections all the way to, let's say, your hand, feet, wherever it is, right? Depending on which vertebral, set these things, the DRG projections are coming from. Okay, now we look at that and we just forget about it. But think about CGRP or substance P that are being synthesized in the DRG cell body, and they have to be transported from that cell body all the way through those extremely long axons for a single molecule or even a vesicle, all the way to the periphery, and then they make effects happen at the periphery. And this boggles me, right? Like how fine-tuned that mechanism needs to be. And if you think about it, it's like a little car starting in Boston and going all the way, you know, past the Bering Strait into Siberia or something, right? That's the distance in microscopic context that is being traveled. How do you fine-tune such mechanisms, right? And all of this is happening in micro scales and in
Yeah.
micro scale timelines. And that just, so these are the things, right, that keep me up and fascinated still and excited to come to work every day.
No, I love it. No, absolutely. I absolutely agree with it. I always find, for example, how quickly hormones work,
Yes.
adrenaline or, any other hormones that bind GPCRs, and then it's you prick your finger and you feel it.
Yes.
And it doesn't take 3 minutes for the signal to travel up. You feel it.
Yes. Yet it needs to cascade through multiple.
Yes.
Yeah, these are the things, right? Exactly. I mean, yeah, we can talk ad nauseum about this. It's just there's so many
Absolutely.
still unanswered questions that give you goosebumps. Yeah,
Absolutely. All right, let's track back because I'm also mindful of the time.
sure.
And do you have a hard stop at the top of the hour?
Yeah, we can go longer.
Okay, good. All right, so you're done with your postdoc. You're co-founding Blue. Think of the audience. Our audience, a large proportion of our audience are PhD students, postdocs. They've worked on GPCRs. What would be your advice to them if they want to go the entrepreneurial route? What to look out for?
It's a very good question. I think I would strongly recommend that every grad student postdoc should get exposure in multiple potential ideas that they can explore for their career growth. We are trained still in academia significantly to be in academia, which is still great. And I think anybody who wants to do that should absolutely do it. But then it used to be, at least when I was going through this, academia or industry. But you're still doing a job, one place or the other. The whole entrepreneurial side, I think, has come to vogue now quite a bit. But still, I think there's significant room for improvement there. So what should be then the mindset, to get back to your question? I think that you need to be clear about two or three things in retrospect. One is, do you have clarity of the question you're asking? Because that's, I think, the most important. Because if you want to go the entrepreneurial side, you have to be solving a problem that is a significant enough problem that in our capitalist society, you can generate revenue for it.
Yeah.
That's the primary point, right? And I know that there may be different stances people might come at it from, but fundamentally, it is the way growth happens. So you need to ensure that you're solving for such a problem, and that problem has a significant proportion of investment that can come in, because otherwise it's a very tough battle, and there's almost very few ways you can make that work. So that's the first piece. Second piece is there are multiple ways to actually fund a company. There's also public granting systems like the SBIR program at NIH, the small business programs at NSF, also the Department of Defense. Different groups have small business, grants. We actually were funded significantly at Blue through such grants as well. And there you can actually bring to bear your academic mindset, and we are all taught to write grants, etc. So definitely hone that skill, right? Because to be able to get non-dilutive dollars is going to be critical. B, what it does is, because it goes through peer review, you also get feedback on where you are, and which can be significantly important. Third thing, and this ties into my previous point, seek feedback. Because I'll tell you, the first business plan that we made for Blue, and we were fortunate enough that MassBio, which is one of the groups that really focuses on small business in Massachusetts on the biotech side, they had a incubation program, and they were inviting 2 to 3 companies each quarter or half a year to come, and they put together a group of 7 to 8 mentors. So we got a stellar group of mentors, VCs, top level pharma executives, etc. And I remember we had sent them our business plan. We walked into the first meeting with them, and then they very politely said, "Please don't mind this," and they ripped up the business plan, threw it in the, in the bin.
Okay,
Right
that's a pretty direct message there.
direct message. It's like, all right, no knock on you guys, but let's start again. Let's start fresh. And what does that tell us, right? So the reason I'm telling you this is you need to take rejection, and you have to take rejection really well, because you will be rejected more than you will be, accepted. And B, you need to have the ability to take that feedback and understand what part of that feedback you can use, what part of the feedback you should use, and what you can ignore. Understanding that is also a skill, and I think that's something you can hone over time. But being aware that you need to do that is the point I'm making. So these are three things. The fourth thing, final thing, is I used to always joke I was chief scientific officer at Blue, but I'm also chief janitor. Like, everything is your problem because it's your company, it's your baby, right? You're starting it. So be very open to that. And during the pandemic, I'll tell you, we had 300 plus fraudulent unemployment benefits that were tried to be claimed on Blue Therapeutics. I woke up one day and there's 300 plus emails from the Department of Unemployment saying, approve all of these. And we're like, no, they're definitely not employees of Blue. So, it's stuff like that. And, you don't have like a significant legal team to kind of deal with it. So you're dealing with these things yourself. So be prepared for such things and take on those challenges positively, because that then gives you the dividend of actually getting the positivity out of the challenges you want to take on. So you have to take the good with the bad. Hopefully that helps. It's a different ballgame.
I think it is, but I think as as a PhD student or postdoc, just being mindful that this world exists is already very important. And now with the internet and ChatGPT and tools like that, if you are interested in exploring such opportunities, you can do it.
Absolutely.
20 years ago, it wasn't as obvious.
It wasn't. I mean, even the programs that I was mentioning were not really well funded or not existing, a couple of decades ago. So this is all, I think this is a great opportunity. And I think I would recommend people actually go and talk to other entrepreneurs that they can get access to. People can reach out to me anytime. I'm very happy to have a chat with anyone for an informational interview, because you just need to understand the battles and see if that's something that is exciting for you. And if it is, you should absolutely do it. I think the world needs terrific entrepreneurs. Absolutely.
Absolutely. In my mind, it's a list, as you know. It's a list. It's organized by date, by time, color coded, and then
yes. Ideally, there's a tracker for it somewhere.
Yes, because I feel like, at least for me, and maybe I'm speaking to a lot of listeners, I need to have a clear picture, of, okay, as you mentioned, four things to remember. Okay, what do I have? But then you need the whole information, and then you put it in different buckets, and that's how I process. You are not like that. You are to the point. Like, you don't see the noise. I need to clear the brush, and it's two different skills. But I think, and I'm getting into our Superluminal adventure here, and I think that's what makes us a good team, is because we see things differently. We have the same goals. We're both excited about GPCR signaling and what it can bring. So let's get into that. Tell us a little bit more about your exit from Blue and how Superluminal came to be, and then I'll chime in with how we came to work together at Superluminal.
Absolutely will do. But I just wanted to again reflect something you said, and I'll get into this, is the number one ingredient is humility, because you yourself will not be able to come up with all the answers, which means you need to surround yourself with the right team. And thank you for mentioning that, because I had not put that as one of the criteria, but that is a critical criteria, because you will not be able to yourself solve all the problems. You need to therefore surround yourself with people who are better fit than you to solve that problem. So that internal humility to know what our skill sets are, and then ensuring, like, yeah, I can be the big vision guy, but if I'm operationally needing to push some of those things, I'll be a disaster, right? Like, I think that's where each of us has our skills, and learning that, understanding that is so critical. So thank you for bringing that up. I just wanted to reflect it.
Absolutely. And let me add something to that, because I think what sometimes people don't think about, or don't, because you're not in that environment. Building a company, it's not a one person effort, it's a team effort. It's like a Lego build out. Everybody brings in their own personality, their own skills, their own colors, but at the same time, the sum of the efforts is it's exponential compared to the sum of the people that bring that in. So I think that's also something to keep in mind.
It is even more critical for anyone coming out of academia, because we typically own the entirety of our programs, usually.
Yeah.
Right? Like, this is how do we talk about it? My project during my PhD, or my project during my postdoc. And we are taught to almost think about it that way, right? It's not that we don't collaborate and whatever, but it's still, I am the first author. The mindset is, that way.
Yeah.
I think when you're thinking entrepreneurial, the mindset has to change a little bit, where, exactly to your point, it has to be, these are the skill sets totally that are necessary. I am bringing this to the table. What else do I need to fill in with, different phenotypes to build that up, right? So absolutely, very, very critical. Very nice. All right. So now going to your question about the transition to Superluminal. Yeah, I mean, as the blue innings, is coming to an end, we had hit the pandemic. It was super hard to raise, for a pain asset. It was, I think, historically, the investment has been less than 2% of all VC dollars go to pain. So it's like significantly underfunded. And during the pandemic, like, we finished our R&D, everything was going great, and we were ready to kind of go. And that's exactly when the pandemic happened. And so it was in the throes of that, we would go and pitch to people, and everyone's like, "Oh, that sounds great. Sounds like a beautiful asset. Do you have anything for COVID?"
Yeah.
So we ended up, like, having to move on from that. And I was pitching different company ideas to various VCs, and I ran into the folks at RA Capital. We had pitched to them as well on this new concept. And one thing led to another, and I got invited to actually, join RA. And RA Capital has this venture arm where they incubate companies, and they bring in entrepreneurs, venture partners, et cetera, so that you can kind of build a team and then foster ideas and then bring that out as a company. So I got invited to kind of join one of those, and Coney was already there, who's currently the CEO of Superluminal. And I think there were some vague ideas about getting into AI and drug discovery. And RA, being a very serious life science investor, they had actually pretty much not made any investments, if I remember correctly, during the hype cycle of AI and drug discovery platforms. In those 2020 to 21, late 21, there was a lot of that happening. They stayed out of that, but they were intrigued sufficiently to say, if you could build a really good life science company that's focused on AI and drug discovery methodologies, how would you do it? And I think that's what, I got pulled into, along with Coney and a few others. And I think what ended up happening was we identified a certain set of areas that we felt could be bridged significantly if we used machine learning and computation at scale, which the current state of the art, let's say, for the biological methods or chemistry methods, are not there. Like, one simple example I'll give you, proteins are very, very dynamic. I mean, think about GPCRs. The conformations that a GPCR goes through for biased signaling, or in the context of, let's say, dimerization, whatnot, is complex. Yet, when we get a structure for a GPCR, let's talk very specifically about it. We first, you need to have a G protein coupled, because there's a certain limit, upper limit on the weight, right? Like, that you need at least this much there, lower limit rather. And you also have to make modifications in the native sequence many times. and also add Brill tags, whatever have you, to stabilize the structure. So you lose that entire dynamism, A. B, you're not even sure what exactly that conformation is. Is that the most therapeutically relevant conformation? So that drove us to start thinking about computational methods. Could you actually teach algorithms, just as with ChatGPT, to understand language? Could it understand the physics of not just folding like AlphaFold does, but could you also teach it the physics of dynamics? What I mean by that is any amino acid in a alpha helix or a beta sheet or whatever has a certain amount of movement it can make in XYZ domains. Now, if you can integrate that across the entire structure, what you get is something like a coordinate density map, if you will, along which you can path trajectories of motion. Along those trajectories, then you can sample conformations. So that's the way we were visualizing it. Turns out you can actually do that. And I think that's been one of the secret sauces at Superluminal, because now you can generate a plethora of conformations. And what does that do in GPCR context? It opens up bias signaling. You can identify agnostically GQ bias, GS bias, GI bias, beta-arrestin bias, et cetera, conformations for any given receptor. And then now you can find ligands that stabilize it, and boom, you're off to the races.
I love it. And I think that's what you just said is exactly what caught my eye and my interest in Superluminal when we were talking about your adventures when it started, because it's such, when you think about it, it's such a simple yet very elegant way of thinking about it. And as you were speaking, I could imagine, everything that you've gone through your PhD and your postdoc come together into here. It's that what if? What are the questions? Who should care? Why should people? How does that help people? And let's forget about what's not possible and let's say, what if?
Exactly.
And I think this is the culmination of all of that training that you got through and all of your experiences.
And the cross-functional thinking, and I think reflecting something you had said right at the beginning, how innovation happens when you have such cross-functional aspects come together, where you're saying, I need to solve for this problem. Let me get over whatever technological hurdles that exist to this date. Now, let's break this down just to illustrate, right? We came at it saying, I want to get as many conformations for any protein. But the problem is, we can only take pictures right now with cryo-EM or crystallography. We don't get dynamics yet. So that's something people are working on. I'm sure we'll crack that. But as of now, we're doing certain things that force us only certain static conformations. Now, what that does, right, is that it completely disables for us the ability to see the entire picture. So we had to go to a different technology, go and see if we can actually flex to machine learning and bring that in, and we can teach that. By the way, when we proposed this initially, the machine learning folks thought I had 10 heads. They're like, that's not possible. How do you even begin to do this? But turns out it can. We've now developed internally 3 different methods to do it. I think that's what it takes. It takes clarity of understanding, having that vision, also making sure that you're not just barking up some random tree. There are inflections you can see, and I think that's where maybe there's a little bit of that intuition as well, to be able to see that and say, I think we're very close, guys. And I think for me, the intuition was, if we can teach algorithms to fold, and actually AlphaFold structures, especially with AlphaFold 2, have been really remarkably good. I'm not saying they're perfect, but you're getting there. I mean, imagine if you had this capability in 2005, what we would have done already, right? So, but then the next frontier is, if you can teach the physics of folding, can you not teach the physics of dynamics? Why not, right? So someone has to push and ask that question. And then you just go that one step further and that one step further, right?
Yeah.
Hopefully that is illustrative.
No, absolutely. You were mentioning about, cryo-EM and X-ray and pictures. And I know you often use the salsa dancer analogy with the movement, but the picture that came into mind is, that school picture where in the morning, you didn't adjust your hair and you put on your shirt backwards. That's pretty much what can happen with a static image of a single conformation of an extremely modified receptor.
Exactly.
You show that to people, they're going to be like, what happened to you? If they know you, others will say, wow, okay, this is you. but that doesn't give the full picture of who you are as a person. And it's the same thing for GPCRs.
Beautifully said. We want to know the GPCRs as they are.
Yes. I often joke, but I half joke about this. I feel like, GPCRs, a receptor in particular, has its own personality in a way, and it all depends on how you treat it and how you tease out the answers from the GPCR.
I agree. It's such a nice, I almost sometimes, I think we've joked about this. I go in, I'm like, all right, receptor X, talk to me.
Yes. What are you going to tell me?
All the secrets.
37 degrees or room temperature incubation? Which one do you prefer?
Oh man.
That's what makes it go. All right, so let's close up because I actually have to get to the office. See, this is what happens when you're juggling all sorts of things. Top 3 aha moments that you had as a scientist that shaped your trajectory.
Oh wow, this is such a good question.
Tough one.
Yeah. So top 3. So one was the first time I actually saw. Let me back up. As you know, heteromerization and homodimerization of GPCRs, I think, still remains a controversial, topic. In Phil's group, actually, he had, predating me by decades, had already shown how when they use these tethered ligands, you were seeing some really fascinating data come out. People can read up about it. Aha moment for me was a single molecule that we were testing, and I could see this beautiful biphasic response, telling us very clearly that this individual molecule was actually binding to 2 sites, a mu receptor and a kappa receptor, opioid receptor, and with 2 different KIs. But allosterically, if you could tether that and put it in, allosterically you would change the KI significantly for the other side, very clearly showing that it was engaging on both of those. And because of the allostery, that there is actual physical coupling going on, which we then later on showed with internalization as well, where you would co-internalize both the receptors together rather than individual and so on. That aha moment blew my mind, because till that point, I had not fully visualized what a oligomer of a GPCR would function like. And it gave me goosebumps. I still remember seeing that data and I was like, oh my God, this is real. So that was one such moment. Another aha moment for me as a scientist was one of the things I did in Clifford's group was, we were working with the TRPA1 receptor, and there was this running joke that anything you threw on the TRPA1 was behaving as an agonist. So I used to joke and say, even if you spit into it, it would activate. Anyway, long story short, what that ended up doing was somebody, Jared Sprague, who was in the group, challenged me. He said, why don't you actually collect spit and put it on, right? So we did, okay. And then we did this experiment where in DRG neurons, we added spit to see if it would do anything. In the same experiment, by the way, after that, 10 minutes later, we were adding capsaicin to all the other wells because we were looking for sensitizers of TRPV1 to see if any of these ligands would also sensitize TRPV1. Anyway, so 10 minutes later into the spit wells, we had also added capsaicin. We didn't think anything of it. Nothing had happened when we added spit. And everybody was like, Ajay, you now have to take your words back. Spit clearly does not activate TRPV1. I was like, stand corrected, guys. I'm sorry. Analyzing the data, Jared comes running to me the next day saying, Ajay, you have to see this. Every well that had saliva in it did not show any capsaicin responses. And we were like, wait a minute, something went wrong. So we did it again, and we did it again. We collected saliva from a bunch. So we, unfortunately, I'm putting this in the public, but, no IRB yet. We had collected saliva from a few different people who shall not be named, and we tried it every single time it worked. And it got me thinking. And that ended up being a big story when I was in Clifford's group. This is why maybe animals lick their wounds, because the one idea that everybody proposed was the antibiotic, antiseptic capabilities. But I know we have a pet, and no knock on pets, but I know my dog at least is not thinking, oh, this is going to become septic in 2 weeks. I need to lick it so that it does it. Yeah. So there needs to be an evolutionarily rewarding response right away. And we propose, could this be an analgesic effect that reinforces that? So you're also lathering on those antiseptic things, right? Turns out that's the case. So if you knock out TRPV1, animals don't lick. And we also identified using mass spec a peptide that actually is a TRPV1 antagonist. It's a native antagonist. So it's a beautiful story. And that, like, again, like I remember telling Clifford the first time, he's like, you did what in our machine?
You spat in the thing.
Yeah, I said big picture, but like maybe not that big picture. Maybe I'll leave it too, because I already taken so much time.
No, I think it's
Probably
fantastic.
Superluminal. The third one is Superluminal. The fact that we could use AI to do the kind of things we're doing at Superluminal it's still, to me, I still pinch myself. I'm like, wow, it's amazing.
Yeah. No, I have to say, I think I do too. Every time I look at our data, I'm like, wow, wait a minute, what now?
Exactly.
And then I call you frenetically, say, look at this, you have to look at this right now, drop everything. Obviously, I'm exaggerating. All right, last question. What should we title this episode?
Oh boy.
I think what came through, let me tell you what came through, and then we can go from there. I think what came through is data is data, or data is king.
Yes.
And also the fact that innovation happens where you set aside or you forget what's not possible.
Yeah.
I think these were the two things that came through.
How about from curiosity to innovation?
Ooh, I love it. There you go. See?
Okay.
All right. Curiosity. I'm taking notes because I am going to forget. Innovation. All right. No, this is fantastic. Thank you, Ajay. I really appreciate you coming. 3 years in, but we did it.
We made it happen. I'm so sorry, but thank you for inviting me. And that said, I'm an avid listener of the GPCR podcast. I've not missed, I think, a single episode, if I remember correctly.
More are coming out. We're starting off, I think, tomorrow, actually. So a lot of cool stuff coming up. Don't go anywhere. I'm going to stop recording, but I want to, get your first impressions after this. All right. Thank you so much, Ajay, again.
Thank you again, Yamina.
Huge thanks to Dr. Ajay Yekkirala for joining us today and sharing his remarkable journey. One of his most powerful reminders is that innovation starts by asking the right questions, not focusing on what's impossible, but imagining what could be done. To stay connected and amplify more GPCR voices, subscribe to the Dr. GPCR podcast and newsletter for fresh stories and insights. Also, consider leaving us a 5-star review on Apple Podcast. Visit terrykinakin.com to explore Terry's Corner and loop in and level up with your pharmacology skills. And check the episode description for links to our partner page to learn more about Celtaris research, their fluorescent ligand tools, and their applications. As always, thank you to the entire Dr. GPCR team for making this episode possible. Stay connected, stay curious, and keep pushing science forward. Until next time, stay safe.